Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled, My Cousin, the Tree, Integrating the Anatomy and Physiology of Plant and Human Biology. I'm Sarah McFarlane, and I'm pleased to be your host for today's event. Joining us today, we're very fortunate to have John Waters, a teaching professor of biology at Penn State, and today he will discuss how his department has developed a plant and animal biology course taken by first and second year undergraduate biology majors that integrates plant and animal biology around common themes. This is the last of the 4 events in the Teaching Anatomy and Physiology series, which has been made possible by 80 Instruments and the Human Anatomy and Physiology Society. So a big thank you to them for their support. For over 30 years, 80 Instruments has been creating simple, flexible tools to help scientists and educators record and analyze data quickly and efficiently. 80 Instrument Solutions brings active learning to your lab, classroom, or online course with easy-to-use software platforms, interactive content, and hardware, including the newly released LT sensors. 80 Instruments customizable education systems provide all the experiments, hardware, software, and lessons needed to teach and create a stimulating learning environment that guides students to connect theory and practice. And so with that, finally, I'm very excited to welcome John Waters. John, it's a pleasure to have you with us today, and the floor is yours whenever you're ready.
Oh, thank you, Sarah. I want to extend my own welcome to everyone here for joining us for the 4th presentation in the 2021 HAPS ADI lecture series. As Sarah said, my name is John Waters, and I'm a teaching professor at Penn State. And I've been a member of the Human Anatomy and Physiology Society for 26 years. But before we begin, I just need to share my favorite HAPS Penn State 80 Instruments tie-in, in that the head of education at 80 Instruments, Ms. Amy Boucher, is a Penn State alumna and a former graduate teaching assistant in the Anatomy and Physiology lab courses that I supervise. So needless to say, I'm very happy to join you today. In this presentation, I'd like to briefly share some of the curricular changes we've implemented in one of our general biology courses, Biology 240, the Function and Development of Plants and Animals. Biology 240 is part of a 4-course core sequence that is taken by all biology majors, as well as students and majors within other colleges at Penn State. This is a 4-credit course where students spend 3 hours in lecture each week and then 3 hours in lab. Approximately 400 students take the course each spring. Today, I'd like to describe some of the changes we've made to the lecture curriculum that we feel have significantly improved the course. A little history. Historically, the course has been taught by 2 faculty members, a plant biologist and an animal biologist, who then divided the course in half. In this format, we were essentially teaching 2 separate courses, with absolutely no integration of concepts between the plant and animal halves. That is how my colleague, Dr. Michael Dan, and I came into the course. I need to stress at this point that everything I'm presenting today grew out of the collaboration between Dr. Dan and I. After working in the traditional format, Michael and I felt that there was an opportunity to make some changes in the course. We wanted to restructure the course to emphasize the interrelatedness of plant and animal biology. By doing so, we felt that the plant biology and animal biology concepts could reinforce one another, and that we could discourage students from creating what we called knowledge silos, where plant and animal biology concepts become isolated from one another. It was also an opportunity to emphasize evolutionary themes, structure and function relationships, and to help students see larger patterns within biology. Instead of relegating plant and animal biology to entirely different parts of the course, we structured our new syllabus around biologic themes that would give us the opportunity to stress similarities between the plant and animal biology. The themes we chose are evolution, movement, structural support, cell signaling, circulation and gas exchange, the acquisition, transfer, and use of energy and other resources, and reproduction and development. Within each of these themes, we still cover plant and animal biology in separate lecture presentations. For instance, The animal lecture about bioenergetics immediately precedes the plant lecture on bioenergetics. So we now have the opportunity to more easily draw parallels between structure and function, or to emphasize evolutionary relationships, having these lectures occur so closely together. So a little bit more about our course. The evolution of plants and animals, and all living things, as I'm sure all of you are well aware, began when the earliest cells arose almost 4 billion years ago. About 1 billion years later, eukaryotes evolved from a prokaryotic ancestor, picking up an endosymbiont along the way to form mitochondria. After another billion or so years, plants and animals diverged, with plants likely gaining chloroplasts through another prokaryotic symbiont. All of this preceded multicellular life on land. So our earliest eukaryotic ancestors may have found themselves floating around in tidal pools along the edges of a supercontinent around roughly 1.5 billion years ago. These ancestors would have shared a complex cytoskeleton with molecular motors, a membrane-bound nucleus, mitochondria, and other membrane-bound organelles, endoplasmic reticulum, mitotic cell division, and some of those cells may have been able to utilize cilia and flagella to propel themselves or secrete an extracellular matrix to form a cell wall. A cytoskeleton of actin filaments, shown in the upper right image in red, is highly conserved among eukaryotes, so it probably traces back to the last common eukaryotic ancestor, almost 3 billion years ago. Additionally, there are scores of myosin proteins, represented in the myosin phylogenetic tree at the lower right of this slide, that fall within at least 17 different subtypes. Okay? These myosin molecules are also found among plants and animals. So whether actin and myosin interact to shorten a sarcomere, as shown in the upper right image, or move organelles through the cell, as shown in the plant cytoplasmic streaming clip at the lower right, myosin motors, which include an ATPase, walk along the actin cytoskeleton to make movement possible. In the bottom left image, you can see that in both plants and animals, these myosin motors are powered by ATP hydrolysis and walk along an actin track. I want to stress here that we do not suggest that muscle contraction evolved from cytoplasmic streaming or anything like that, but rather plant and animal cells both inherited an actin-rich cytoskeleton and myosin motors from a common eukaryotic ancestor, and then each evolved mechanisms that utilize those elements to create movement within the cell. It is comparisons like these which we believe help students focus on larger patterns. This way, the plant biology and animal biology each reinforce one another, helping our students form more complex connections between individual concepts. While some prokaryotes can secrete a cell wall, the cytoplasmic machinery in plant cells allow them to secrete a cell wall utilizing cellulose. And as my HAPS colleagues know, animals can secrete a variety of substances to create different forms of extracellular matrix. For instance, if we compare the cell wall of a general plant cell, shown at the upper right of this slide, with the bone cells below, you will notice that both secrete an extracellular matrix that includes polysaccharides, such as pectin and cellulose in plants, and mucopolysaccharides in animals, as well as proteins. With the addition of other components, such as the calcium phosphate and calcium carbonate in bone, both plants and animals can form an extracellular matrix that resists compression, but also maintains a degree of flexibility. Evolution likely selected efficient arrangements of this extracellular matrix in both plants and animals independently. For example, both osteons and tree trunks take advantage of how well columns resist compressive forces to support animals as large as dinosaurs, as well as trees that may weigh several tons. Our students are certainly aware of the use of columns as architectural elements that support buildings, but they often overlook biologic structures, like tree trunks and osteons, that are also organized into columns. The discussion about extracellular matrix in plants and animals, then, is another opportunity to discuss the relationship between structure and function. For instance, if we start with a pair of columns, one with a round cross-sectional area shown at the upper right versus one with a square cross-sectional area shown at the upper left, and then I begin to load each column with compressive force, in this case, my granddaughter's books, the column with the square cross-sectional area will collapse first due to a weak point halfway between the corners, which make the square column more prone to buckling. However, as you can see, the column with the round cross-sectional area, similar to osteons and tree trunks, is much more resistant to a compressive force because that force is distributed more evenly around the circumference of the column. However, like bone and tree trunks in real life, any column that can eventually buckle if enough force is applied. So the formation of a stable extracellular matrix likely played a role in the evolution of multicellular organisms and presents another opportunity to compare and contrast the relationship of structure and function in plants and animals. Moving toward a new topic, circulation, unicellular organisms and other plants and animals with a high surface to volume ratio can rely on diffusion between their cells and the external environment to exchange gases, nutrients, and waste. However, larger plants and animals have insufficient surface area to rely on diffusion as the only mechanism of exchange with the environment. These larger organisms have independently evolved transport mechanisms to transport nutrients and waste and allow us to observe additional commonalities between plant and animal structure. In physiology, we teach our students that the volume flow rate of a gas or liquid is proportional to the pressure differential, or more simply put, gases and liquids will only flow if there is a pressure gradient. There is no flow without some sort of pressure gradient. Students are fairly familiar with this idea in the context of the circulatory system. No matter if it's a two-chambered pump connected to a hollow body, as found in arthropods and mollusks, as shown at the left of the top diagram, or a four-chambered heart, more like their own, that utilizes circuits of arteries, veins, and capillaries, as shown at the right, students understand that if the heart does not generate pressure, then flow of the circulatory fluid will stop. But then, given that plants do not rely on any sort of pump, how is a tree capable of generating the pressure differential necessary to move water maybe dozens of meters from the roots up to the leaves? During this section of our course, we would still cover animal circulation and plant transpiration in this example separately, but we use the need for generating a pressure differential as a unifying theme between the two. As you may recall from your own general biology courses, while a small amount of root pressure develops as a result of active transport and osmosis in the root tissue, that is not enough pressure to move water up the stem of most plants. However, during transpiration, water evaporates from pores in leaves called stomata, shown at the lower left, and as water molecules move or evaporate from leaves into the atmosphere, a pressure differential gets generated in that region. Through a combination of cohesion, which is the attraction of water molecules to one another, and adhesion, the attraction of water molecules to the wall of the xylem, the tube containing the water, the entire chain of water molecules lined up in the xylem is pulled upward from the roots, as shown in the figure at the lower right. This is also an opportunity to prompt students to compare and contrast the role that tubes play in the transport of materials through an organism, another structure-function tie-in, whether the arteries, veins, and capillaries of animals, or if we're talking about the xylem and phloem in plants. We can take a similar approach when we discuss the flow of fluid out of and into a capillary bed versus the movement of sap from the leaves of a plant to the roots through the phloem. A pressure differential, hydrostatic pressure, drives both processes. In an animal circulatory system, The top figure illustrates how a hydrostatic pressure at the arterial end of the capillary bed, generated by the heart, creates the pressure differential that pushes fluid through both the lumen of the capillary and simultaneously pushes fluid out of the capillary and through the interstitial space as it washes over the cells. The lower figure illustrates that in plants, a pressure differential also pushes fluid from one region of the plant to another, in this figure, down the phloem and toward the roots. In addition to hydrostatic pressure, we can also use this opportunity to discuss osmosis. In animals, hydrostatic pressure drops as blood flows through the circulatory system, so much so that the proteins that remain at the venous end of a capillary bed create an osmotic pressure that exceeds the hydrostatic pressure and draws fluid back into the capillary. In plants, the sugars that are produced in the leaves via photosynthesis are pumped into the phloem, which creates an osmotic gradient that pulls some of the water out of the xylem and into the phloem. This increase in phloem volume helps create that hydrostatic pressure again, that pressure gradient that drives fluid flow through the phloem. Again, we are taking the specifics of fluid transport in very different organisms to illustrate larger concepts associated with fluid flow. Cell signaling is another area where we can reinforce unifying ideas. Anatomy and physiology students are often familiar with the gap junctions that connect the cytoplasm of adjacent cardiac cells, but then they're surprised to discover that plant cells have similar structures called plasmodesmata. Both connections allow the diffusion of intracellular messengers, ions, and other material between cells. These intercellular pathways can also transmit local potentials from cell to cell, allowing both plant and animal cells to communicate with one another. While the first multicellular life may have been an algae-like ancestor 1.5 or so billion years ago, multicellular plants and animals existed at least 600 million years ago. Both inherited genes used to regulate proteins and membranes and likely independently evolved mechanisms for cell-to-cell communication. We've always spent a lot of time in our anatomy and physiology classes and in the animal physiology portion of this course, teaching students how nerve and muscle cells can generate and propagate rapid changes in membrane voltage via the ion channels associated with action potentials. What students often do not realize is that action potentials also occur in plant cells. The Venus flytrap is a classic example where insects are attracted to the plant's color and nectar, and the inner portion of the leaves of a Venus flytrap contain between 3 and 6 trigger hairs, shown in the photograph in the upper right. These trigger hairs act as mechanoreceptors. If an insect touches 2 of these trigger hairs, or if the same hair is touched twice within about 20 seconds or so, an action potential is generated which causes the leaves of the trap to snap shut, trapping the insect. These action potentials also seem to stimulate the release of enzymes that then digest the insect. Both plant and animal cells also use a variety of chemical messengers to communicate between cells. Glutamate is one such messenger. It is the most abundant excitatory neurotransmitter in the central nervous system of vertebrates and has also been found in plants where it seems to be released in response to stressful stimuli, such as herbivory. The stimulation of glutamate-like receptors can trigger a calcium signal that can travel from plant cell to plant cell via the plasmodesmata and may initiate other biochemical pathways in response to stress. So again, we can draw parallels with calcium as an intracellular messenger, with local potentials, with the cytoplasm of cells being connected to each other. And I think it makes all of this much more relevant to these students that kind of have a bias toward animal biology and especially toward human biology. Signaling throughout the entire plant may also be possible via action potentials, which have been shown to travel along the phloem of some plants in response to some stimuli, almost like nerves. It's been proposed that these action potentials may be integrated with other plant signaling pathways as the plant responds to stimuli. It is unclear exactly which ion channels are involved in plant action potentials, but the fact that plants and animals both evolve these signaling pathways is another way that the concepts from plant and animal biology can reinforce one another. So plants and animals share this common eukaryotic ancestor that incorporated an endosymbiont, which formed mitochondria, and sometime later, plants incorporated that second endosymbiont that became the chloroplast. Mitochondria and chloroplasts both utilize electron transport chains in ATP synthase. So whether we first cover plant or animal bioenergetics, the physiology of one provides students with prior knowledge to more easily understand the physiology of the other. Plants and animals both use mitochondria to transfer high-energy electrons obtained from organic molecules down an electron transport chain. That's shown at the top of this slide. That generates the hydrogen ion gradient, which is really an electric current, which supplies the energy to ATP synthase to build ATP from ADP and phosphate. The streaming chloroplasts we observed earlier use a similar mechanism where electrons excited by light energy are transported down another electron transport chain and also provide the energy needed to generate their own hydrogen ion gradient, which powers the ATP synthase in chloroplasts. We believe that emphasizing these common themes makes our pre-medical students a little less likely to dismiss plant biology. In the previous version of the course, when plants and animals were taught separately, the students treated the plant portion as something to be survived before they got to the good stuff, the animal stuff. All right? But this way, we think the students are less likely to dismiss the plant biology, and we're reinforcing central concepts across all organisms. We stress similar commonalities when we cover plant and animal reproduction and development. We still spend entire lectures discussing the specific details of plant reproductive biology and separately animal reproductive biology using model organisms. But by placing both the plant and animal lectures together within the reproductive and development portion of the course, we can emphasize and reinforce common themes such as the challenges faced by both plants and animals as they moved from an aquatic environment where the transport of gametes, maintenance of cells, and nourishment of the embryo were more straightforward, and then transitioned to land where desiccation, gamete travel, and embryo nourishment became much more of a challenge that were solved by the evolution of seeds in plants and amniotic eggs in animals. During the lectures on plant and animal development, we also use pattern formation as one of the organizing themes. Students observe that plants and animals have an organizational structure, and we'll even spend time in class to ask the students to list the organizational patterns they see in plant and animal images that we project on the screen. For instance, plants have leaves above the ground and roots below. Animals have a head on one end and a tail on the other. Furthermore, the leaves of plants have an adaxial or upper surface and an abaxial or lower surface, just as animals can have a dorsal surface and a ventral surface. This is another case where plants and animals used their genetic eukaryotic toolbox to independently evolve different developmental pathways. Plants use Madsbox genes to regulate the development of their body plan, while animals use Hox genes to do the same. Both types of genes are present in both plants and animals, but they code for different regulatory proteins. The important thing in our course is to get students to think about the patterns we observe in plant and animal body plans and how the structure of the organism as a whole, as well as its individual parts, determine the function. So we set out on this path because students did not seem to like learning plant and animal biology as a divided curriculum very much. For that matter, the instructors teaching the course did not like the course very much either. Teaching plant and animal biology during separate halves of the course seems sort of like a shotgun wedding, or maybe at best, that a spoonful of human A&P helps the plant biology go down. In the divided curriculum format, student comments were often negative about the plant portion of the course. As I said, they'd survive it. They'd they'd put up with it until it got to the animal part. And overall, the course received low ratings on our end of semester course evaluations. Now, to be honest with you, the first time we taught the course using this integrated curriculum, the students hated it. It was horrible. We still took a team teaching approach that semester, and in the written comments, the students told us that they perceived the course material as being extremely fragmented. They weren't sure what to study and when to study it. Fortunately, at Penn State, instructors are encouraged to take risks, and we understand that good things may be happening in a course even when the course evaluations seem very low. The second semester that we used the integrated curriculum, Dr. Dan took over the entire course, and he smoothed the rough edges and made the plant and animal connections much more explicit. And that was really, really important. Students appreciated his work and reflected that both in their comments and their overall evaluation of the course. So after going through this, we feel that our experience demonstrates that integrating plant and animal biology does not need to feel like that shotgun wedding. The integration of processes in organisms that seem very different at first glance provide opportunities to emphasize evolutionary relationships, how structure determines function, and even how physics defines how organisms solve biologic challenges. After we worked out the kinks, students appreciated this approach, and the faculty teaching the course are much happier to be involved in the lecture portion of the course. I just want to say a few thank yous. Oops. Sorry, there we go. I'm very fortunate to work with a large group of faculty at Penn State who prioritize undergraduate education, and I am extremely thankful to work with Dr. Michael Dan and all of the faculty and staff in the Department of Biology and the Eberly College of Science at Penn State. I also want to thank the nice people at 80 Instruments and the Human Anatomy and Physiology Society for making this series possible, and a special shout out goes to our host, Sarah McFarlane at Insight Scientific, for bringing all the tech aspects together today. And most especially, I want to thank everyone here for joining us today. I feel so fortunate to work in a profession with so many talented and dedicated colleagues from literally around the world. After the recording of this presentation is saved, I've included a few references at the end that may be helpful. I've also included credits for the many, many people who share their artistic talents with educators like us. So thank you very much, everyone.
Wow. Thank you so much for that, John. Really fantastic presentation. I know you're bringing a lot of themes together for people who have been through those undergrad courses. So thank you again for that. All right. So to kick off this Q&A, the first question here is, John, you mentioned that a 1-credit lab is part of this course. Can you tell me how you've used this approach in the labs?
Yeah, good question. We haven't used it much yet. Yeah, I was very aware that I did not discuss the laboratory portion of the course today. We're actually just beginning those discussions about how to redesign the laboratory portion to integrate it with the new lecture format when the pandemic hit last year. And then everything went out the window, and like everyone here, we all went into survival mode. One thing that grew out of the online laboratories that we developed for this course during the pandemic was that it provided an opportunity for us to develop at least some case studies, if not online resident instruction labs, but case studies that aligned very nicely with the integrated lecture portion of the course. And now, as we're getting ready to transition back into in-person instruction, we've started those conversations again to see where we could use those case studies to build upon a hands-on laboratory experience that integrates with lecture. So that's actually still a work in progress.
Okay. We have another question here for you, John. This person has said, it sounded like you said that your administration at Penn State forgives bad course evaluations. Can you tell me more about that?
Yeah. the forgiveness is not infinite. You can't have bad course evaluations forever. But there is a recognition here, and one of the reasons that I feel very thankful to work here is that in order for instruction to improve, and in order for faculty to improve as educators, in order to improve our courses, we have to try new things. We have to take risks. And it's possible here for instructors to let the administration know, hey, I'm trying something new. This is why I'm doing it. This is what I'm doing. And you kind of get a pass on any negative SRTEs are the course evaluations that we do. You get a pass that semester anyway on any negative course evaluations that may be the result of you taking this risk. And then the whole idea is that, no one wants to see a course in trouble across time. But the whole idea is that you take those comments and then try to make it a little bit better the next semester and a little bit better the next semester. We had one course that's in that 4-course sequence that the course evaluations, after they did a profound change in the structure of the course evaluations took, I think, almost 3 years to bounce back to their previous levels, and now they're much higher than they ever were. So sometimes you got to take a risk and kind of tough through the bad stuff, and that's built into the system here.
That's awesome. Okay, we have another question here. James has asked, was that all in one semester, and was there time for non-plants and animals?
Let's see. Most of the changes that I presented here today, we had implemented, yes, in one semester. The course is only taught once per year, so we had basically and it's taught in the spring, so we had a summer and a fall to do this design. Now that said, it gets tweaked every single semester, and I really got to give credit to Dr. Dan for just making constant improvements in the course. But yeah, we kind of pulled the trigger and did almost all of that. That change from separate plant and animal biology to this integrated approach from one spring to the next spring. Whether or not we do non-plants and non-animals, not so much. I mean, this is specifically a function and development of plants and animals course. The prerequisite course includes a large biodiversity component, and that's where students get introduced, even to other plants, to algae, protists to prokaryotes, and things like that. This course tends to focus, for plants, tends to focus mostly on land plants, on angiosperms and gymnosperms. And even though we use different animal models, say to illustrate different reproductive strategies or different types of respiratory physiology or whatever we happen to be covering, we have so many pre-meds in the course that there are a good number of references to vertebrate and even human biology as well.
That's fantastic. All right, keep the questions rolling in, guys. We've got lots of them to go through, but we have lots of time, so that's great. We have another question here, but it's a comment and a question. So thank you. I'm excited to adapt your approach to my own majors course. I assume that this course has been offered online recently due to the pandemic. Do you have students work through online labs on the plant physiology portions? Because he has had a hard time locating any.
Yeah, there were some the online labs for this semester were primarily case studies in plant and animal biology. So, whether or not that's in the spirit of a real lab, and that lab is so hands-on, no, we're really stretching, the definition of lab. Last spring, when we got hit with the pandemic right during, fell right during our spring break. There we adapted some plant labs, but it was a wreck. We were doing this microRNA lab and how different microRNAs are expressed due to stressful stimuli. And it's a pretty cool lab online. It didn't transfer, or it's a pretty cool lab in person. It didn't transfer to the online format at all. So even though we developed it, we just got rid of it. And then, as I said, we used case studies sort of in lieu of lab as our lab this year that aligned with them better. But the case studies really don't have a very hands-on component. And that's something that we're working on now between this spring and spring of 22.
Okay. Someone else has asked, did you create a textbook for the integrated course?
We've talked about it. We've tried a couple things. We've tried using just class notes. We've used notes and articles and websites and things like that. We did a custom published version of a traditional general biology book where we selected just the chapters that that were relevant, and none of them have worked out that great. We had looked when we started to see if there's a textbook that takes this approach, and we didn't identify one. There might be one now. Since Dr. Dan took over the course, I've been working on some other projects, but we have talked about creating a textbook. I think it'd be actually really cool. Again, it just takes time, right? 24 hours, 365 days a year. That's what we all have. But I think something like in an OpenStax format would be amazing. Or maybe, and I'm just brainstorming, as my colleague Kevin Petty says, just friends talking. Maybe taking some of that material and rather than reinvent the wheel, reformatting it in this, with this approach might be, might be useful.
Yeah, for sure. That'd be great. So we have another question here that is, what are the prerequisites for this course?
So as I mentioned, this course is part of a 4-course sequence. We used to do a biology 101 and 102, but we felt that there were that there are too many foundational concepts that needed to be covered that we could fit in reasonably into a 2-semester sequence. So we moved to a 4-semester sequence. The very first course is a foundation in biology course, which covers Darwinian evolution. Biologic chemistry, just kind of a quick review. Darwinian evolution, a little bit of population genetics, like Hardy-Weinberg kind of stuff, origin of life, Mendelian genetics, and then a big biodiversity component. That is the course. That course serves as a prereq then for each of the 3 other courses. The 3 other courses can actually be taken in any order. But the other 3 courses in the 4-course sequence then are an ecology course, a molecular biology course, and then the course that I described today, the function and development of plants and animals. So there's only one prereq for our course, that first one.
Okay, great. Sounds like a good group of courses to be in. Another question here from Peter, which I will actually answer if that's okay. Loved the figures. Where are they from? In John's slides, which will be uploaded along with the resources we just talked about, he's got a ton of reference slides, so you'll be able to click through those once the webinar is on demand. So hopefully that answers your question, Peter.
Yeah. I can add a little bit of that, if that's okay, Sarah.
Yeah, go ahead.
Yeah, almost all of those were from Wikimedia Commons. So yeah, all of them are, have Creative Commons licenses or public domain licenses. I'm sure that a lot of you have had the same experience that I have had this year. in developing online resources and being told by your university that you're not supposed to violate copyright. So we developed some experience finding images online that we could legally use, and I'm amazed at how generous illustrators have been with their artistic talents by sharing these through Creative Commons. So that's where most of them came from, and I have links to each individual one that, as Sarah said, we'll post.
Great. So we've got another question here from Karen. Karen has said, in our introductory biology course for majors, it's taught as your original model, and one common student complaint was the difference in exams and teaching style between co-professors. Not necessarily that one is better or worse, but just different. How do you handle exams and grading?
Since Dr. Dan took over the course, that's become a moot point, because now there's one instructor that does the entire course. I am involved in some other courses. For instance, our Anatomy and Physiology 1 lecture during the fall, we run 2 different lecture sections of that, and the faculty involved in those courses meet every week and actually write exams together. It takes a bit more time, to do that level of coordination, but it has been a nice way to address the students' frustration in, maybe it's a reality, but maybe sometimes it's just the perception that they have 2 different courses because there's 2 different instructors. But the only way that we found to do that is through a lot of coordination with the instructors.
Awesome. Collaborative efforts for those assessments. Okay, so we have another question here from Anne. Anne has asked, did you often employ assessments which required students to integrate their knowledge of plant and animal functions through applications of some kind, like written explanations?
Yes. I didn't really talk about the assessment portion of this, but you have good instincts, Anne. The redesigning the assessment part of this course was also a huge effort. Writing questions that capture both, that push the students up more toward that application and analysis level of thinking has taken a lot of time too, and really is a continuing effort. Mostly through multiple choice questions in our course, because it's so large. Like I said, we have 400 students in a single lecture meeting in a single lecture room when we're resident instruction. A lot of it has been just to try to write good, high quality, higher order questions like that. That's another place where the pandemic actually made life a little bit easier, because relying on the online testing and the whole academic integrity issue is an entirely separate question, but putting that question to the side, we could write a greater variety we could use a greater variety of question formats through our course management system than we ever could through a paper exam.
Great. Alrighty, we've got another question here from Ryan. He's asked, how do you handle different terminology? For instance, turgor pressure versus hydrostatic pressure. Do you introduce both or try to simplify?
We introduce both, because this course then is serving as a prerequisite for the 400 level out at our university. Majors courses go from 100 level through 400 level. And this course serves as a prerequisite for 400 level courses where they are going to use that more specific terminology. So both in a plant physiology course and in a human pathophysiology course, we need to cover both so that they can make that transition.
Right. Okay.
Sorry, I had a brief moment of panic when my screen went up.
Oh, that's scary.
Everything's good.
Okay, great. Dee has asked or has said, John, talk to me about how we create interactive, self-directed learning modules similar to a textbook but interactive using ADI's LT platform.
I have not used ADI's LT platform yet, so I can't be that specific. I'm guessing that you've spoken with a colleague that I've been co-teaching with about interactive, self-directed lessons. And that's been another kind of positive thing, I guess, that grew out of this pandemic experience, where we started. We've been creating online lessons, actually built in a upper level a medical physiology course that I'm teaching, built around Dee Silverthorne's textbook, where we structure every week's lesson using a self-regulated learning model, where we try to engage the students first with something about that topic relevant to their to their prior knowledge, to their experience, to real life, and then use that as a transition into learning the material. That's where something might be more text-based, whether it be a OpenStax book or working through the assigned textbook, with guided questions and things like that, and prompts to study diagrams or to make tables or draw things and things like that, and then take them from that learn stage to an application stage, where they take this information that they've just learned and then try to apply it to something new. This is where we would introduce a case study, or perhaps a figure from a research article or something like that, something they haven't seen before that they're going to have to digest and chew on, and then kind of close the circle to bring them back to everyday life and real life and their own personal experiences. We end with a reflection section where we ask them to take this experience and how has their view changed? How has their understanding changed? How do they feel about some topic? I wrote one about what was it? In the one on mechanics of breathing, I wrote a case study based on the experience of a friend of mine who was a emergency department technician, who helped treat and sadly witnessed the death of a college student from alcohol poisoning. And, so they were in the apply section, we were talking about the effects of alcohol on the rate of breathing and what that would do, to blood chemistry and things like this. And we could talk about acidosis and hypercapnia and all these cool things. But then we brought it back to alcohol poisoning and, what can you do to prevent it if you were ever in this situation where you saw someone in distress? And it was, a really cool circle to close. So that's the approach that we've been taking, trying to develop self-guided interactive lessons. We've not used that model for Biology 240 currently. This semester, that course was taught more as a synchronous online lecture. But it is an approach that we're using in a number of our courses that we're really enjoying, and so far the feedback from the students has been pretty good. Sorry, that was a very long answer.
No, that's okay. That's all right. All right. So to respect everyone's time, we're going to only take one more question for today. But if you do have any other questions for John, please submit them using that Ask a Question panel, and we'll make sure to forward those along to him. John, this last question says, thank you for sharing, first of all. And then her question/comment is, I have had the same difficulty of the negative perception of students in general towards plant biology/physiology. Yeah. They view it as not interesting or irrelevant. How do you address this difficulty?
I mean, in a nutshell, that's why we went to this integrated approach. And what we see on the course evaluations is much of that resistance has gone away. Among the various teaching and learning theories, there's various ideas about what motivates students. And in one model of student motivation, they focus on two things: that the students find the information useful, and that they also find the information, they find the information useful and relevant to themselves, and that they believe they can be successful in learning this thing. And I feel that by taking this integrated approach, where we talk about the role of a positive pressure, be it turgor pressure or hydrostatic pressure, in moving fluid through a tube to nourish an organism, we're helping the students see that utility, that relevance, and how learning the plant parts help you understand the animal parts better, and vice versa, and hopefully give the students a better understanding of biology as a whole. That was our whole motivation for doing this, and so far it seems to be working pretty well. None of the pre-meds are deciding to become, plant biologists. It's not that successful, but that level of resistance, or that rolling of the eyes that we would see sometimes about plant biology, this seems to have helped that quite a bit.
Yeah, that's fantastic. Okay, well, I just wanted to thank you again, John, for all of your fantastic insights today, both in your presentation as well as the Q&A session.
Oh, thank you. It's been my pleasure.
Yeah, we love having you. And I also wanted to thank everyone for joining today to attend the webinar and taking time out of your day. I know everyone's super busy. The other 3 events in the series are now all on demand, so don't forget to check them out if you missed them. We also wanted to thank our lovely sponsors one more time as the series comes to an end. Thank you to the Human Anatomy and Physiology Society and also to 80 Instruments for making these events possible. We've gotten so much positive feedback about all 4 of them, and we're really happy to be able to produce them for you guys. So thank you again for taking part in this webinar and the whole series, and we look forward to having you all with us again soon.